Okay. Um, One more thing to talk about here is, well, yes, you can have an abnormal number of chromosomes, just holistically as a whole chromosome, but there can also be alterations in chromosome structure. You can be mixing pieces of chromosomes or missing or adding extra pieces of chromosomes, and that can also affect the outcome of the cell and the organism. So here are 4 examples of what can go wrong with chromosome structure. So here we have a typical chromosome, has sections of genes A through H. And you can have deletions occur in chromosomes. This is where a section of the chromosome has literally been removed. And so you miss all the genes that are found in that section of chromosome that's been deleted. You can also have duplication. So in this case, you'll notice segment B and C here has been repeated twice. And again, in this case, you would have extra copies of all those genes that are located in segments B and C. And so for both deletion and duplication, it's pretty obvious that you're changing the gene dosage. You're having either too many copies of certain genes or too few copies. And again, this imbalance creates problems for the cell. We can also have what is called an inversion. So in this case, we've taken segments B, C, and D, and we've left them on the same chromosome in the same spot, except we've flipped it around, so mirror image. And while that doesn't seem so bad, right? You still have the same number, copy number of those genes. But something we haven't talked about is that genes oftentimes have regulatory sequences that are way upstream of the beginning of the gene. And these regulatory sequences, when we talk about them later in chapter 17, or chapter 18, they help to control how frequently that gene is expressed. So how much of that protein do you make, for example? And so the genes here in section B might have their regulatory elements in section A. And if you flip that segment of the chromosome around, you might be separating the genes in section B from their regulatory elements in A. And so now all of a sudden, maybe those genes are no longer expressed. Or maybe they're overexpressed. And so while you don't change the gene dosage, you do change how much of that product is produced, again, creating that imbalance for cells, which can create problems. And then lastly, we have an example here of a translocation. So translocations are when you take 2 non-homologous chromosomes and you swap parts of the chromosomes. So here we're flip-flopping AB with M, N, and O. And again, doesn't seem like that would be too big of a deal because you're not changing copy number, but again, you might be separating genes from their regulatory elements. And just to give you an example, in humans, there are certain leukemias that result from a translocation between chromosomes 9 and 22. When you flip-flop parts of these chromosomes, it can lead to cancerous conditions. Okay. And then the last part of this chapter is taking a look at 2 more examples where inheritance patterns deviate from simple Mendelian genetics that we've been studying in chapter 14. One of these examples has everything to do with genes located in the nucleus of a cell. And the other example has nothing to do with those genes. It has everything to do with the genes that are located in organelles like mitochondria and chloroplasts. But in both cases, in the nuclear example and the organelle example, in both of these examples, the parent from whom the genes were inherited makes a difference in how those genes are expressed, right? So it says here one exception involves genes located in the nucleus, the other exception involves genes located outside the nucleus. But in both cases, the sex of the parent contributing the allele is a factor in the pattern of inheritance. So let's take a closer look with some examples. For the nuclear gene example, we're going to walk through this process that's called genomic imprinting. So in this case, it matters which parent passed on a certain gene to you. Because for some genes, for some mammalian genes, and we're still learning a lot about this process of genomic imprinting, the gene from one of the 2 parents oftentimes gets silenced. and is not expressed. And so if the parent that gave you the mutant gene gets their gene silenced, then it's not a big deal in terms of your phenotype. But if it's the other parent that passed on the mutant gene to you, then you're going to be expressing the mutant gene and not the healthy one, and that's going to be a problem. So again, let's take a look at an example. So this example is in mice. And we are looking at a specific gene called the IGF-2 gene. This is an insulin-like growth factor. So it helps the animal to grow, right? It's a signaling factor that will cause cell division to increase mass of this animal. And so this IGF-2 gene, it has a normal allele and also has a mutant allele. If you get the mutant allele, then you will be smaller in size than you would if you express the normal allele. And so this top mouse right here, this mouse has inherited 2 normal alleles. So a normal one from the dad and a normal one from the mom, no problems there. Now, for this particular gene though, what we have determined is that the allele that you inherit, or the mouse, inherits from the mom, it always gets inactivated. So the maternal gene never gets expressed. So the mice only express the allele that they get from the father. And so these 2 mice, now this is where the problem arises. These 2 mice are heterozygotes. So they are genetically identical for this gene. They have 1 mutant copy and they have 1 normal copy. But you'll notice one mouse is big, or let's say normal size, and the other one is teeny tiny, as a dwarf mouse. Same gene, same genotype, but different phenotype. How did this happen? Well, remember, you get 2 copies of this gene, but you only express the one that you get from the dad. So this particular heterozygote got the normal allele from the dad and the mutant, the dwarf allele, from the mom. That's not a big deal. You don't express the allele that you get from the mom, and so only the normal allele for the IGF-2 gene is expressed. This mouse is normal sized. This poor little guy, though, he got the normal allele from the mom and the mutant allele from the dad. Well, for the IGF-2 gene, the allele received from the mom does not get expressed, and this mouse only expresses the mutant allele and is therefore then a dwarf-sized mouse. All right, and then the last example, again, has everything to do with organelle genes, or what we call extranuclear genes or cytoplasmic genes. And these are typically found in mitochondria in animals or in plants. They can also be found in chloroplasts and other plant plastids or other organelles that can contain small circular DNA molecules. And what we have determined as scientists is that extranuclear genes are inherited solely from the mom. So all of your mitochondrial genes you inherited from your mom. Your dad didn't contribute any. Now, we don't know the exact mechanism behind it. Maybe the sperm doesn't transfer any of the mitochondria, or maybe the mitochondria from the sperm are actually degraded. We don't 100% know yet. But regardless, the zygote only contains mitochondrial DNA from the mom and not the dad. So that means that if your mom has a metabolic disorder where there's a mutation in her mitochondrial genes, your mom is going to pass on that mutation to all of her children, because all of her children get all their mitochondrial DNA from the mom and not the dad. And the first evidence of this extranuclear inheritance really came from the study of plants. I'm sure you've seen plants out in nature, or maybe even your backyard, that are variegated, meaning that they have like a blend of colors, maybe white and green, or maybe pink and green. That inheritance pattern of the pigments in the leaves actually comes from extranuclear genes that are inherited from the female that contributes genes to creation of the seed. And so you can see it here in the really beautiful coleus plant that sometimes we use in lab for various experiments.